this script F is the set of amplitude frequency pairs of the complex conjugate eigenvalues. So just to sort of dig into what these different terms mean, we can break down this P of t into stationary states, monotonically decaying states, and damped oscillating states. So we can actually show that if a distribution or a SAT problem does not have these damped oscillating states, then the optimization problem is actually convex. It's much easier to solve, whereas sort of these damped oscillations in the continuous time evolution tend to result in a significant increase in the amount of time required for a solver. So suppose we sort the eigenvalues of this P matrix in descending order, and then we can sort of estimate the number of steps required for a SAT solver, given by this formula here, with respect to this value lambda k, which is the first eigenvalue that does not have modulus 1. And we can see that as this value approaches 1, we sort of see a quite rapid increase in the estimated number of steps required by a SAT solver. In fact, this is on a log scale. So you can see that the closer this lambda k value is to 1, the longer it takes to find satisfying solutions. So now we're going to transition to some models and experiments that we performed. So ultimately, there are two quantities that we want to approximate with respect to the spectrum of this matrix. First of all, we want to know what is the value of this lambda k, so we can estimate the number of steps it would take to find a solution. Something also that might be useful is the stationary distribution, corresponding to some initial state, which is just the tensor power of a randomly selected initial state. And to do this, we'll propose a simplified surrogate for P called a belief propagation network. So a belief propagation network, or BPN, is a stochastic block matrix that has this form, where sort of this, for example, B++ is the probability that a variable, in this case, x sub j plus 1, propagates its value to another variable x sub I. And you see we also have analogous definitions for the other elements of the block matrix. So for example, if we have the following sentence, we would obtain this matrix as our belief propagation network. And so here's just a visualization of the spectrum of this BPN. Again, note that what we're doing here is we're taking a 2n by 2n matrix and using it to approximate a 2 to the n by 2 to the n matrix. So obviously there is a lot of information loss. We can see that the spectral gap, the difference between 1 and the first non-unit eigenvector is approximately 0.6. And on the right, you can see an Argand plot of just the eigenvalues of this belief propagation matrix. So again, this is symmetric because the eigenvalues come in conjugate pairs. So we can account for the noise parameter of these different stochastic algorithms by sort of augmenting the diagonal of this BPN matrix. And what we can do is we can sort of plot what is the principal eigenvalue, the first non-unit eigenvalue, and how does that vary with the clause to variable ratio of a problem. And we see that as we increase this clause to variable ratio, adding more and more constraints to the problem, we see that if we don't add any noise, then the problem actually converges faster. But the reason that happens is because it actually converges to local minima. Whereas if we add noise, then the problem can actually escape those local minima and converge to a global minima. But that actually results in a significant increase in solver time as we increase rho. One other thing we can do is we can also extract information about the stationary distribution using this formula here. So we can sort of estimate the probability that a given variable has a particular assignment, assuming that satisfying solutions exist, by basically taking the corresponding entries in the stationary distribution of the belief propagation network. And what we did here is we computed the mean Hamming distance of an optimal most probable satisfying assignment. So we took all the set of satisfying assignments and we sort of computed what's the probability that a particular variable is true or false in those sets of satisfying assignments. And then based on that probability distribution, we made sort of an optimal maximum likelihood guess as to what that satisfying assignment is. And what we noticed is that we can actually almost guess around about 75% of the variable assignments just by looking at the stationary distribution of this belief propagation network. One thing that's worth noting here is you sort of see this general trend where it increases and then decreases. This point where the cost to variable ratio is around 4.3 is actually what's called the phase transition point. Maybe we could talk about that, but it looks like about running out of time, so I'll just hop to the conclusion and say that by interpreting stochastic stat solving as a Markov process, we can approximate the following. So we can approximate the norm of the first non-unit eigenvalue, lambda k, and we can also approximate the stationary distribution. And so this model, by preserving the dynamics of unit propagation, can produce quite reasonable estimates of these values in polynomial time. So I guess I'll open up the floor to questions. And these down here are my references.
Awesome. Thank you so much. Do we have any questions from the judges?
Looks like Denton.
Yes, go ahead, Denton.
I'm sorry, I can't find the hand raising feature on here. This is great, Colin. Thank you so much for all of this. You didn't really get into what are the implications of doing it this way as opposed to the many other ways that have been proposed for stat solving.
Yeah, that's actually a great question. So one of the problems that happens when you start increasing the scale of stat problems is that traditional methods like conflict-driven clause learning, they're what are called complete methods, meaning they give you a definitive answer, yes or no, the problem is satisfiable or not. Whereas stochastic algorithms will tell you with 100% accuracy, yes, this problem is satisfiable, but if it gives you a no, then there still is a chance that there could exist a solution. So one of the things that's useful about stochastic algorithms is they're used more in the context of physics and statistical mechanics, because more often it's useful to provide a probability bound that a problem is unsatisfiable versus producing a 100% probability guarantee of whether or not it's satisfiable or not. So in order to actually provide that 100% accurate guarantee, then you have to do an exponential amount of work.
Okay. Awesome. All right. So just to stay on time, we'll move on to the next presentation. Okay. Before we begin, judges, do you need any more time finishing feedback? Okay. We'll give like one more minute. Okay, awesome. All right. Our second presentation is by Siddharth from Texas A&M. You may share your screen and begin when you are ready.
All right, are you all able to see my screen? Okay, cool. Right, my name is Siddharth Pandian, and I'm a senior computer science student at Texas A&M. And my research is about integrating external learning applications into learning management systems, or LMS, which is in the domain of HCI. So with online learning becoming the new norm, learning management systems such as Blackboard and Canvas play an increasingly vital role in higher education. This is shown in LMS's 24% annual growth rate. Currently, the top 3 LMSs are Blackboard, which holds around 31% of the market share, and Canvas, which holds around 30%, and Moodle, which holds around 18%. However, recent trends are showing that Canvas is actually going to overtake Blackboard due to its better grading functionalities and third-party application integration. Despite all of these advances in LMSs, many instructors tend not to use these new technologies due to many reasons. Previous research by Gautreaux and Lawler-King show that these reasons are closely related to demographic data, such as instructors' age, gender, department, etc. On top of this, instructors tend to use multiple learning applications to aid instructions. For example, a chemistry professor may use Cengage OWL or ALEX or Pearson Learning, and a physics professor may use Mastering Physics in order for students to better grasp the material. The main problem with having so many multiple third-party applications is that it becomes cumbersome to manage and consolidate every single student's grades. Transferring grades from the application to the learning management system can prove to be time-consuming and inefficient, depending upon the specific third-party application. So each third-party application has its own way of connecting to the LMS, which is not ideal. Multiple standalone applications also pose the problem of managing multiple login and passwords. We've all experienced like if you forget your login and passwords for multiple different websites, it's kind of annoying. So, and also managing due dates on each platform may become confusing as due date changes may not be clear to students. So my research has 2 main goals. One is to understand instructor motivation. This will help us to understand how much instructors really care about third-party software. Also, we can learn about specific features about educational software that instructors may find most important. It may also prove beneficial to understand a student's interest in third-party software. To accomplish this, I created a comprehensive survey for instructors and students that were based on motivation theories. I'll explain these motivation theories and the survey design later. The second goal of my research is how do we integrate a standalone application into an LMS? So what are the modifications we can make to a third-party application in order to fit in or integrate seamlessly into an LMS? And how do we implement the specific features that the instructors care about in integrated software? The solution to this is Learning Tools Interoperability, or LTI. LTI is a standard developed by IMS Global Learning Consortium. It is a standard for integrating any third-party application into any LMS, whether Canvas or Blackboard. This will allow students to seamlessly switch from many different LTI apps with ease. Basically, LTI solves the problem of integrating cloud-based learning applications with learning management systems. The technical implementation of LTI will be explained later as well. By gauging the instructor and student interest in integrated software, we can possibly provide like some sort of avenue to make third-party applications more widely adopted through the use of LTI. So now onto the motivation theories. So what motivates an instructor to adopt new technologies? The first theory is motivation hygiene theory. So according to Herzberg's motivation hygiene theory, there are 2 main factors for job satisfaction: motivating factors and hygiene factors. Motivating factors are factors that directly impact your motivation, such as recognition and praise, fulfilling work, etc. Hygiene factors, on the other hand, are factors which could possibly demotivate you if you do not receive them, but do not directly impact your motivation. These factors include pay, working conditions, and administrative practices. If you do not receive your pay, it may possibly demotivate you to work. This study of Herzberg is particularly useful because proper survey questions can be crafted that targets this theory. Administrative practices is one of the main hygiene factors that is relevant to this research. Therefore, questions regarding the role of university practices in influencing an instructor's decision to adopt an LMS can be asked in the survey. The second theory is called the diffusion of innovation theory, developed by Rogers, who is a professor of communication studies. This theory pertains to the rate of adoption of new technologies. According to this theory, there are 5 main categories of people. We have the innovators, the early adopters, the early majority, the late majority, and the laggards. As the name suggests, the innovators are people who adopt a new technology first and the laggards are who adopted the last. The following figure from the study by Rogers portrays these category of peoples versus the market share of the new technology. Since LMSs are a relatively new technology, the diffusion of innovation theory still applies. And LTI is an even newer technology, therefore it's even more relevant. The hypothesis is that faculty who are innovators and early adopters are going to be the most likely to favor LTI apps. Therefore, we need to identify some of the demographic data of faculty members who are early adopters of new technologies. Questions about age, tenure status, gender, department, computer proficiency, salary, and history are all relevant. Now onto the survey design. Most of the questions that I ask in the survey are Likert scale questions, which are basically questions that make you rank your feelings about a certain topic from 1 to 5. These questions ask about the subject's sentiment toward integrated software versus non-integrated software. A sample size of 30 was chosen because it's the ideal sample size to perform statistical analysis since the central limit theorem holds. Currently, the survey is still in the process of gaining IRB approval and should be obtained soon. So here are some questions that I ask in the instructor survey. I first ask questions that collect some demographic data on the professors. This will be useful in creating our groups or samples for statistical analysis. And these questions target the motivational theories. As you can see in the top right question, we ask about the extent to which the university has encouraged the use of LMS. And this targets the motivation hygiene theory. And the other questions target the diffusion of innovation theory. So by ranking the computer proficiency, we can estimate who is a laggard versus who's an early adopter or innovator. So a person who picked 5, who's very proficient in computers, is probably going to be an early adopter or an innovator, versus a person who picked 1 is probably a late adopter or a laggard. And here's the meat of the survey. Here I ask questions about an instructor's sentiment about using non-integrated software. The keyword is not here. And then I ask the same mirrored questions about integrated software. So here I show an example of integrated software. So this right here is the Mechanics platform, which is a third-party education software created by the Sketch Recognition Lab at Texas A&M. And here's like a sample sketch of this integrated with eCampus. And then I ask the same questions that I ask about non-integrated software here, about integrated software. And the reason I do this is because I'm using a paired sample t-test, which is essentially taking 2 measurements from a single group. So the most common example for paired sample t-test is like, say you want to administer like an exercise program to a group of, to a sample. And you first measure the before weights of the people, and then you administer the exercise program, and then you measure the after weight. And then you see if there's any difference between the before and after. So our experiment is analogous to this. Our before is the instructor's sentiment on non-integrated software, and our after here is our instructor's sentiment on integrated software. So the null hypothesis is that there is no difference between the instructor's sentiment on non-integrated and integrated software, and the alternate hypothesis is that there will be a difference between these 2 sentiments. And then after this, we can perform a 2-sample t-test, which is 1 measurement from 2 groups. So the 2 groups here can be instructors versus students, or instructors within different age brackets or academic rank or department. So to better illustrate the design of my experiment, here we have the instructor samples, and then we have the student samples. So within the instructor's sample, each instructor is asked a set of non-integrated questions and integrated software questions. So it gauges their interest on these 2 subjects. And since this is taking 2 measurements from a single group, we use a paired t-test. And the same can be done for students. We use a paired t-test between the non-integrated and integrated questions. And then once we do this, we can then use a 2-sample t-test to measure the difference between the 2 groups, the instructor and students. So we can just take these, the integrated questions from the instructors and the integrated questions from the students, and then compare those 2 means. since it's only one measurement from two groups. Now, the beauty of this experiment is that we can be flexible. So our groups can be anything. So within the instructor group, I can split the instructors into early adopters and laggards, and then use a 2-sample t-test within them. And this can be anything. So I can change the early adopters to professors who are from 30 to 40 versus professors who are from 50 to 60, and then use a 2-sample t-test to compare the means. Now this is the second goal of my research, which is the technical implementation details of LTI. So before we go into this, we must first ask, what is standard LTI? LTI is a single sign-on assertion, or SSO. In other words, it's a one-way directed identity. So it is a way for someone to go from the LMS to some other web service already logged in. For example, the credentials used in Canvas will be passed directly to a third-party web application. A predefined key and secret is shared between the tool and LMS, and there's a sign-in process that happens. The predetermined key and secret is shared between the LMS and the app. A launch happens, which is basically a post request, which sends user information to the app and also signs that request so that the tool knows that it's from the LMS and not from anywhere else. The app can then send back content such as grades and stuff back to the specific user, or even auto log in the specific user specified in the form post. So the LTI documentation uses the term learning tool consumer and learning tool provider, but in my mind it becomes kind of confusing. So the learning tool consumer is basically the LMS, and the learning tool provider is the third-party application. And so each third-party application has an XML app configuration, and in this app configuration you specify the endpoint to your third-party application that can accept post requests from the LMS. And then an app launch happens, which is basically sending the post request with the student's credential to auto log in the student. So so that's some information about the technical implementation details. And this is quite useful, is because like each education software has its own way of connecting to an LMS. So the LTI kind of solves this problem because it's a standard. So say a third-party software is being tailored for Canvas. So maybe 5 or 10 years down the road, if Canvas is no longer the most popular LMS, and if it's say Moodle or Blackboard, then there's more work to be done to modify the third-party application to fit the other LMS. So an LTI is basically the standard. So even it's basically future-proofing the third-party application. So that ends my my presentation. So I'll open the floor to any questions.
Okay, any questions for the judges? Okay,
yes,
go ahead.
I have actually, please. Cedar,
Yes.
I would like to know more about the application. This is an application to implement in the website, or application for example, on the smartphones or other kind of devices. Could you explain more about?
Yeah, so it's any sort of applications that can accept post requests. So it could be mobile, it could be web applications. So yeah, it's basically open to. So this application that I showed the example here is Mechanics, which is basically it's a web application, but it also has yes, it also has a mobile component to it. So it really depends. You can use both mobile and web applications.
Okay. And thank you.
Yeah.
Good presentation. Thank you.
I did see, Denton, that you have a question, but unfortunately we're running low on time, so I'm so sorry. But are all the judges done with their forms, or they still need more time? Yes? Okay, awesome. So our third presentation is by Hodis from UT Austin. You may share your screen and begin when you are ready.
Thank you. May I ask if anyone can see my screen? Thank you. Hi everyone, thank you so much for having me and giving me this chance to present my research. My case study will be analyzing how to measure heart rate using the Ppg technology. But first we need to know what is Ppg. Ppg is photoplethysmography. Photo means graph and plethysmography means any changes in volume. Therefore, photoplethysmography can provide us information about any changes in volume and be able to enable us to graph it and see it visually. One of the uses of this technology is to measure any changes in blood volume through the Ppg sensors. PPGs are, we can also define them as non-invasive and optical measurements that we can use the source of light to reflect any changes of blood flow through the amount of absorptions of light in our body organs, for example, tissues. PPGs has the same function as electrocardiograms or EKGs, but with the simpler procedures. And there is a slight variation from the actual heart rate when we use Ppg technology, but that can provide, therefore we can conclude that we can provide the reliable measurements using these measurements during COVID-19, or we can validate the heart rate count at the, and compare it with the much lower compared commercial alternatives and use it as a screening tool for unfacilitated communities. Ppg can also provide us information about the individual's age, any cardiac diseases, and irregular heartbeats. Next, we're going to see how we can use and get the graph of Ppgs. We're going to use the Circuit Playground Express. These are really accessible, and the reason that we use this Circuit Playground Express is easy to work with, and it's can be provided at low cost. Here we can see that this part is the light sensor, and here the Circuit Playground Express has 10 lights that can be turned on. However, we only use the first light, or the second one. It's up to us, and light it and use the sensor by putting the finger on the sensor and measuring it. Here is the overall step of the procedure that we're going to use. This is the code that's going to be programmed on the Circuit Playground Express. So Circuit Playground Express is going to be connected to the computer or laptop, and we can program the Circuit Playground by, for example, shining only one light or two lights. Therefore, we only shine one light, and it's important to note that there are multiple types of lights, for example, infrared lights, green LED lights, or purple lights. However, green LED lights shows the higher amount of accuracy and absorptions of light through fingers. Therefore, we use green LEDs, and we set the adjust the brightness to the maximum level of 255 when we want to work with the Circuit Playground. Afterward, when we put our fingers on the sensor, it can provide us this Ppg Signal Graph, which can be shown here. So here is the light level over the time, and we can see that here, as the blood flow increases, it's gonna show the systolic peak, and here it's gonna show the second peak, gonna show the diastolic peak. And as I mentioned, Ppg can provide us information about any irregular heartbeats or cardiac diseases or even the age. So here I provide the examples that we ran on the two people. This is the graph of one 40 years old man, and here it's about 90 years old child. We can see that the distance between the systolic peak and diastolic peak are much narrower as the age increases. And here, for the distance between systolic and diastolic are much wider. That's why it can provide us also information about the age and validate that. So next, after we got our Ppg graphs, we're gonna use FFT, FFT is fast Fourier transforms. And in order to calculate the or analyze the PPG, we need to have PPG data on the Excel sheet. And it's also important to know that we can run FFT on multiple platforms. For example, R, MATLAB, Excel. In this case, I used Excel. And in Excel, we're going to have 3 columns to calculate the FFT graph, to get the FFT graph. Which the first one that is important is FFT frequency, FFT magnitude, and FFT complex. It is also important for us to have the information about sampling frequency. Sampling frequency is the number of data points or the samples that we have in 1 second. Therefore, the way that we calculated sampling frequency is that we counted our total sample sizes of data points, which was 256, and we divided it by the total amount of time, the time that it took for us to measure all the data points. In this case, sampling frequency was about 18.29, and using the sampling frequency information, we can get the FFT complex, and FFT complex enable us to get the FFT magnitude and FFT frequency informations. What we need to care and we care about in this analysis is FFT frequency and FFT magnitude, since it's going to be shown on our Y and X axis. After graphing it, it's going to provide a graph that looks like this. All the FFT graphs have the similar shape. Usually they have a peak and they have a really lower Y coordinates as the frequency goes on. But in order to count and calculate the heart rate, we need to use the peak of the graph. So here we can see the coordinates of the peak is 1.07 and 10.113. And since we want to get the heart rate, we want to use the frequency to be able to get the number of heart rate in 1 minute. So we look at the X coordinate of this peak, which is 1.07, and we multiply it by 60 to give us the information about heart rate. So heart rate in this case is gonna be 64. And after we run the PPG, the actual heart rate of the 40-year-old man was 62. So these measurements was really close to the actual heart rate that enable us to see that these are reliable. We also perform more samples on child also, and also at a different time and the 40-year-old man. We can see that here the X coordinate is 0.999, and we can, by multiplying that by 60, which is number of seconds in 1 minute, we can get the heart rate of 59.94. In this case, the actual heart rate was 61. Still, it was about 1 or 2 points of difference variation. And also the heart rate count was 1.195, and multiplying that by 60 gave us the heart rate of 71.7, which is pretty normal for a child, since children have a higher heart rate compared to adults. And it's also important to say that PPGs have the same function as electrocardiograms, but working with a simpler procedure. What we hope to achieve in the future is try to implement a procedure on the more data and gather more samples, and also add on more features such as SpO2, heart rate variation, respiratory rate to the prototypes. PPG has a lot of functionalities and important features that we can use to gather information regarding vital signs. And as I said, it's functioning very similar to electrocardiogram, and it can be used to monitor the heart rate of patients when they are going to the operating room and they are under anesthesia, to constantly monitoring their heart rate. So that's one implication of that, of the PPG technology. And here I need to thank Freshman of Research Initiatives at CNS, College of Natural Science at UT Austin, and my research educator, Dr. Timothy Rydell. And these are my references. Thank you. Here I open the floor for any questions.
Awesome, thank you, Hadis. Any questions from the judges? Yes, Denton, go ahead.
Oh, that was just applause, but I do have questions. So in the medical field, obviously, error measurements have to be very, very small because we are dealing with human life here. And so you did talk about the 2-point difference between the measurements that you have there. What kind of error analysis are you doing, and how close are you all to something that would be medically significant?
Absolutely, that's a great question. It is really important to minimize the amount of errors and standard deviations in medical field, as it has a really high implications if it has errors. Therefore, we just try to validate the heart rate count and to see if we can provide that during a real emergency conditions, such as for unfacilitated communities or during COVID-19, that people can get these screening tools compared to the very higher expensive tools. And in the matter of analyzing the errors, we run how there is a 1 or 2 points of difference or standard deviation between the actual heart rate. However, we did not analyze the errors in more depth since that was the 3 months of research project, and basically the time constraint didn't allow us to look more into those details.
So the goal of this was to just get something out fast so that a bunch of people could use it outside of medical facilities.
To get it as accurate as possible. And there are multiple ways of doing that. We just try to use the circuit playground because they are they can be provided at a very low cost. For example, we can also use Arduinos and sensors and microcontrollers. Those are also provide us a better measurements, and they are more accurate. Plus that they have the oxygen saturation level embedded in them. But yes, this prototype is prone to errors, but a small amount of errors. And it's important to address all of those errors when we test them on different people.
Awesome, thank you guys. Now, before we move on to the next presentation, do the judges need any more time finishing writing their feedback? No? Are we good? Okay, awesome. Okay, so our fourth presentation is by Sam from Baylor University. You may share your screen and begin when you are ready.
Can everyone hear me? Yep. Okay. All right. Hello. So my name is Sam, as was mentioned, and I'm excited to be here with you today to present my research, load testing bipartite graph visualizations, a machine learning approach that was completed under the supervision of my advisor, Dr. Eric Baker. So here's a little bit of background about the project and kind of the impetus for why we decided to do this work. So GeneWeaver is this website that is currently online, is currently available, and it allows users to answer a lot of biological questions using genomic data. And so the way that it goes about doing that is by first allowing users to upload gene sets of various genes to GeneWeaver for analysis. So these can be genes collected from GWAS studies, from genome-wide association studies, and from whatever genomic source is available. And then users can then take these gene sets and group them into what we call projects in GeneWeaver. And then finally, and perhaps most importantly, analyze these gene sets using the analysis tools present on GeneWeaver. So some examples are the Boolean algebra tool, the Jacquard similarity tool, and the tool that we use mainly for this work, which is the hierarchical similarity graph. So a little bit of background about the HiSim graph or hierarchical similarity graph is that it results in a hierarchical network of bipartite graphs representing gene set interactions. And so just for those who are unfamiliar with bipartite graphs, a bipartite graph is a set of graph vertices that can be split into two sets such that no two graph vertices within the same set are connected by an edge. And so the plots that we decided to implement, these improved visualization, are called the BZ subway plots, and they represent an improvement to the current visualization implementation. And we can see here on these graphs on the bottom, the graph on the left is the current existing GeneWeaver graph visualization for the HiSim graph, and the graph on the right is the improved subway plot visualization. And as you can see, like the graph on the right is a lot prettier. It's more colorful and allows you just to kind of see the flow and the ebb of their gene sets a lot more effectively than the graph on the left. And I think I'm sure as all budding computer scientists, we all know that people love pretty pictures. And so I hope that's like the takeaway from my presentation, at least, is that we want to make sure people are happy and they get the pretty pictures that they want. So the problems that happened with this work was that the VC plots themselves were not previously implemented in GeneWeaver. So they were a library that was separate from the GeneWeaver ecosystem and implementation. And they didn't really fit in with the existing GeneWeaver implementation. And they also had been shown to not be able to handle displaying graphs for some reason. And so we really didn't know like what graph size caused it to not display correctly. Was there some other features that didn't allow it to be displayed correctly, and so on and so forth. And so those were the two main challenges that we had to address in this work. So that kind of brings us on to the methods. So in order to solve both of these problems, we first implemented the VC plots in the GeneWeaver ecosystem. And so that we did that by kind of modifying it so we allowed them to connect to each other and display correctly. And then secondly, we load tested the visualization using two different machine learning approaches. And I'll talk more in detail on these two approaches in the next few slides. But just for reference, we can see here kind of the workflow of our two visualization or our two machine learning approaches. I called the first one machine learning approach 1 and the second one machine learning approach 2, which I know is a very unique name. And so to do this, really, to be able to run these approaches, we needed data. And I'm sure all you machine learning people know that we can't really do machine learning without data. And so to do this, we had to generate two data sets. The first data set we generated was from random runs of the Hygiene graph, which we called the real runs. And so what we did here was we found, oh, these are all the gene sets that are publicly available on GeneWeaver. We're going to randomly pick gene sets and throw them into the Hygiene graph and see what happens, right? And sometimes we'll get like a nice, big, pretty graph. Sometimes we wouldn't get any graph at all. But that was the idea. It's just run the Hygiene tool with random gene sets and see what we get. And we'll store those graphs that do work as one data set. Then we also took 10 of those graphs that we used to this approach, and we randomly permutated them. And so what this did is it redrew the edges between the nodes without changing the number of nodes or number of edges. And the real idea for doing this is because we wanted to make sure that there was no latent factors, there's no within the structure of the graph that was causing the visualization to incorrectly appear. So for instance, if we had a graph that we knew displayed correctly on the VC plots, but then we permutated it and it didn't display correctly, then we knew that there was some difference in some latent feature that we couldn't observe in the graphs that would cause it to not display correctly. So then our second data set, where we were able to collect more data, was graphs that were randomly generated based on node, edge, and hierarchical depths. And we call these fake runs. And so these are just randomly generated graphs. They weren't run through the Hygiene tool, and they just mimicked the output of the Hygiene tool, but they were fake. And then we manually annotated all of these graphs in order to determine, did they appear correctly on the VC plots or did they not appear correctly on the VC plots? So then that gets us into our first machine learning approach. And so our first machine learning approach, we used a graph autoencoder, abbreviated as GAE, in order to try to discover any of those latent features inherent in the graphs. And what graph autoencoders do is they take our giant graphs and they reduce the dimensionality of it, and they reduce it to a form that we can compare easily between multiple graphs. And that's probably a high level explanation of what they do, but for the purpose of this work, that was what we were focused on. And we used 4 different types of graph autoencoders. We used the GCN AE, GCN VAE, linear AE, and linear VAE. And for the sake of time, I'm not going to go into the differences between the 4, but just know that we tried 4 different approaches. And then after we got those graph autoencoded graphs, we then used a PCA dimensional reduction to reduce that 2D output of the graph autoencoders to a 1D representation so that we could then cluster the graphs. And then we use a k-means clustering approach in order to do that. So in our second machine learning approach, called machine learning approach 2, we used a random forest machine learning algorithm, and we trained this based on permutations of the following factors. So n, which is the number of nodes, m, which is the number of edges, max d, which is the maximum depth of the tree, density, mode d, and the number of self loops. And you can see here on this graph on the right, the run number and which factors were used in, or which factors were used to train the graph for our different runs. And so we trained our random forest on the real random data that we had collected, and then we tested it on the fake runs. So that gets us into results. So as first, we were able to implement the VZ plots in GeneWeaver, as you can see here on this picture on the right. So people got their pretty pictures. I'm sure they're very happy with that. And then what we did is for our data generation, we collected 78 real runs. And again, what as I kind of alluded to earlier, is not all the graphs, when we ran the HiSim graph using randomly picked gene sets, worked. And this was because there might have just been no overlap between the genes. So if you had a gene set that was for, let's say, alcoholism, and you had another one that was for Behçet's disease, which is an autoimmune disease, the gene set overlap between those two is very small, if any. And so that's kind of the reason why a lot of the trials that we ran for these real runs ended up failing, and we were only able to collect 78. And then of those 10 graphs that we selected for random permutation, of which I think I have 5 of them displayed here on the bottom, 2 of them were not able to be displayed. So this was confirmed our initial suspicions that there's some graphs out there that we cannot visualize using the VZ subway plots. And so as you can see here, all these are real runs, and there's a pretty wide spread in how the graphs look and how many nodes they have and how many edges they have. And so that we were able to get a pretty nice distribution of the number of nodes and edges and representation of the graph using this random approach. And then secondly, we collected 1,725 fake run graphs. I don't have any of them displayed here, but we collected all of those. So then for our machine learning approach 1, unfortunately, embedding the graphs resulted in poor performance, and we couldn't continue along with this work. And this was despite any technique used. So any of the 4 different types of encoders that we used, we were unable to achieve significant results. And I didn't show the data here because it was that bad. So then for our second machine learning approach, we had much better results, and they were much nicer and much cleaner. And our random forest classifier was able to classify correctly those fake runs 88% of the time when using just n, m, and maximum depth. And here on this graph on the right, you can see which features were used and what was the score that the classifier was able to obtain using just those features. And what we can notice here is when we use nm and maximum d, pretty much for whatever additional feature we use, our results are pretty high, like they're pretty consistent. But our best result was just only using those 3 factors, which was good because it's a lot easier to test graphs when we're trying to display them based on those 3 factors than it is to find the density of a graph before we display it. So then our final prediction, we did this. We used that classifier to determine what plots can be displayed correctly, and we found that the VZ plots were able to handle graphs that had around 50n, 65m, and 30d. And the way we got these numbers is we had 2 approaches. The first one on the left here, we plotted in green which were the graphs that worked, and in red which ones didn't work. And if you look at based on visual inspection, you can see it's around 50, 55, and around like it looks like it's around like 1,000 or 1,000 amp, which doesn't seem correct, right? And then our depths here we can see on the top there. But then because that one's a little, we aren't able to see it as well, we decided to then plot it in a histogram and then take this 1 standard deviation ahead of the mean, which is how we got these final numbers. And you can see here on this node graph, 1 standard deviation is around 50 n, 1 standard deviation of the edges is around 65 m, and then 1 standard deviation of depths is around 30d. And so that's how we ended up with our final numbers. And so I think I'm running out of time here, so I'm going to just briefly say that this is an improved visualization experience for GeneWeaver. And then the failure of these graph autoencoders to encode might just be due to the fact that these graphs that we tested were too small for the encoders to work correctly. And then just some acknowledgements and my works cited. And I'm happy to answer any questions that you may have.
Awesome. Thank you, Sam. Do we have any questions from the judges? Yes, Dr. Arduz,
Yes.
go ahead.
Yes, thank you, Samuel. It was really good and interesting presentation. My question is, did you use autoencoders and PCA algorithm, right?
Yeah.
Both of them. But why we should use both if just one? If we can reduce the dimension just with one algorithm.
Right. So that's a great question. And so the use of the graph autoencoders is that we have these nodes and these edges in a graph, and it is able to deconstruct that graph into a 2D representation. And the main use for that is pretty much determining edge relations within the graph itself. So it's not really used to compare across multiple graphs, right? It's mainly to determine something within a graph itself. So it doesn't reduce it down to one dimension, which is what we can use for our clustering analysis, which is why we had to use PCA.
Okay. Yeah. Thank you.
Okay, great. Oh yes, Radu, if you want to go ahead and ask your question.
Hi, Samuel. Good presentation. Yeah. So I have two questions. The first one is a follow-up to what he just asked.
Okay.
I was wondering what would happen if you reduce the dimension to just one dimension for your autoencoder, and you use that for the clustering. How would that compare with doing it to the dimension you reduced it to before reducing it to the PCA? And then number two, I think it's a classification problem, right? So I wanted to know how a simple linear classifier would fare against the autoencoder, because I think you are comparing it for several graphs.
Right.
And so how many graphs did you use for the data, and how would a simple linear regression do?
Okay, so I think I'll start with your first question about reducing it down to one dimension. We noticed that the issue wasn't so much with the dimensional reduction as it was with the encoders themselves, right? So despite like which parameters we use for the encoders, our results are still really, really bad. And a lot of the graphs that we tested weren't even able to be encoded using the autoencoders, despite like which parameters we used, right? So I think our PCA reduction step, when I tested it, it was like 95 to 100%, which I know like might seem like overfitting. But when I talked to Dr. Baker, it was like, just because we're going down to one dimension, the PCA isn't the limiting factor there. I don't know if that makes sense.
Yeah.
And then your second question was, I believe, based on what we trained our data with. And I think I had a slide here that shows how much. So we had 78 real runs is what we used to encode to train our graph. And these were 78 randomly generated graphs. So they had a pretty wide spread, I think.
Right, thank you very much. I think, well done, well done.
All right, thank you.
Okay, awesome. So do the judges have any, do the judges need any more time to complete their forms? Good, all right. Okay, so our fifth presentation is by Ashley from the University of Dallas. You may share your screen and begin when you are ready.
Hi, my name is Ashley Nguyen, and I'm a student at the University of Dallas. I'm going to be discussing our research over a machine learning approach for identification of potential antiviral compounds against SARS coronavirus 2 structural and non-structural protein targets. SARS coronavirus is the virus responsible for the 2019 COVID-19 pandemic. To date, there are about 120 million cases and 2.7 million deaths worldwide. To find a solution, we took a machine learning approach to utilize unsupervised and supervised machine learning techniques and apply it to 60,000 molecules of interest for this target virus. K-means was an unsupervised learning algorithm used where the model was able to find patterns in the data that allowed it to group similar observations into groups or clusters without being given a specific target variable. In contrast, supervised machine learning using random forest and R created a model that predicted the value of a specific target variable based on the observation's other data. And to compare the two, one emphasizes clusters of similar data points while the other focuses on individual classification or regression predictions. The goal of our research was to identify antiviral compounds as inhibitors of SARS coronavirus 2 to aid in the development of potential drugs and treatment. Our hope is that our research will aid in decreasing the infection and mortality rate referenced in the previous slide. To begin our research, UT Southwestern took a pharmacoinformatics approach to curate 49,431 antiviral compounds with antiviral activity or structural similarity to known antivirals from the CAS registry using Constance Information Miner. In order to identify potential compounds as possible inhibitors of the virus and establish a set of controls to be compared against the CAS dataset, a literature search was performed to identify antiviral compounds with potential activity against COVID-19. And 32 control variables were then identified through calculating molecular descriptors and fingerprints done by UT Southwestern. A K-means algorithm was applied to the data obtained from UT Southwestern to determine the ideal number of clusters to classify the data. And K-means specifies the number of clusters K initialized centroids by shuffling the data set and then randomly selecting the K data points for the centroids without replacement and then continuing until there's no change to the centroids. An example of the output of K-means clustering is shown in the image to the right. And after running the algorithm, it was determined that 2 clusters were ideal to use as they had the highest silhouette value. The silhouette score measures how well the data fits divided by the number of clusters, and the higher the silhouette score, the better the data. The algorithm was then ran on the 32 control molecules to test if they fell into the same cluster, concluding to have similar known effects on treatment. Next, a comparison was conducted to look at the decimal fraction of COVID-19 drugs in cluster 0 and cluster 1. In comparing the 2 frequencies, as shown on both these graphs, the 2 frequencies of the control and experimental molecules, overall, most of the molecules were found in cluster 0. The molecules found in cluster 0 provided evidence that the molecules have a therapeutic effect and are more similar in their properties. All clustering from this point was to ensure the accuracy of the data. And despite these findings, there are possibilities within the data that K-means does not consider into the data, such as various potencies based on molecular environment or different drugs targeting the same receptors. Therefore, we then turned to random forests to enable the tuning of the data as our research progressed. The 32 antiviral control molecules identified earlier on as having physiological and therapeutic effects, but compared to a data set of 25,573 experimental compounds that have not yet been studied. The Kennard-Stone algorithm was used to pick out 32 representative molecules to create the training data set consisting of 32 control molecules and an additional 32 experimental molecules to apply to the experimental compounds. The training data set was then analyzed using random forests, which is a method that builds multiple decision trees and merges them together to get a more accurate and stable prediction. The assessments were executed on the training data set, with the target variable being the status variable, which classified molecules as either experimental or control. To increase the model's accuracy, 7 of these relevant variables were removed from the training data set. These variables were identified by generating the relative importance of features plot, where variables were plotted according to their mean decrease Gini, measuring the variable importance. The plot over here shows the measure of relative unsaturation content relative to molecular size as the most important classifier of potential antiviral compounds, where the x-axis denotes descriptors across each of the experimental and control molecules. The figure shows the relative importance of each variable in classifying the training data points. As shown in the figure, more important variables contain a higher decrease Gini. So with the increased variable importance as you move further on the graph. The adjusted model was then used on the testing data set consisting of 23,757 remaining curated molecules from the CAS registry. And a receiver operating curve was calculated measuring the area under the curve of the classifier, which showed a 92% predictive ability of the compounds. And based on the calculations, the classifier identified 178 compounds as potential inhibitors for the virus. To summarize, the model developed aimed to identify molecules of interest for treating SARS coronavirus 2 from a group of unstudied molecules based on chemical data of control molecules that have been previously identified to have potential positive effects on treating COVID-19. And after finding 178 potential antiviral compounds, the data was then communicated back to UT Southwestern for further analysis using multi-molecular docking analysis. And to further our research, we would like to search for more potential molecules of interest as well as new control molecules, in addition to conducting more specified clustering methods, such as agglomerative clustering, which would potentially draw circles and loops in between data, making clusters as dense as possible. And all codes utilized for the assessments can be found and accessed through our GitHub as a reference or used for further experimentation. This concludes my presentation. Thank you all for listening, and I'll be happy to answer any questions.
Awesome. Thank you for that presentation. Are there any questions from the judges? Yes, Paul, go ahead.
Ashley, well done, Ashley. I wanted to find out, you had 2 classes for your random forest, right? All right, so would you say that it's somewhat consistent with the number of clusters that you had using the k-means, and then finding out that the silhouette gives 2 as the number of groups, as the best group, best k? Would you say it's somehow consistent?
Yes, well, we're targeting at 2 ideal clusters, and then after running the algorithms, we found that the control molecules ended up falling within those 2 clusters where they fell specifically under cluster 0 and then cluster 1.
Sorry, can you repeat? I didn't get you.
Oh, so there is a correlation between the data obtained with ideal clusters. Like, for instance, in our research, we had 2 ideal clusters for the controls. And after running the algorithms, they fell within 2 controls. And specifically in our research, they fell under cluster 0 and clusters 1, where I believe 29 out of the 32 molecules fell under cluster 0.
Okay. All right. Thank you very much.
Okay, Denton, do you have any questions?
I am curious. So you say you were able to get it to about 78 different drugs, which may or may not be able to help with the COVID-19 positive results. What kind of, you mentioned this a little bit, but what kind of ways do you further vision to get that number down even further?
Well, conducting our machine learning experiments, we were able to narrow it down to 178 compounds. And then we took that number and then we kind of passed it along to UT Southwestern, where they conducted molecular docking to narrow that number to 78. But in terms of improving, I guess, our research for later on to adding to the molecules, I would say, like I said on my last slide, conducting more specified clustering, such as the agglomerative clustering, that closes or increases the density between the clusters without allowing space. Like, I think of it as a gray space that we aren't able to determine. So just closing that gap.
Got it. Have you considered any approaches other than clustering, simply because this seems like more of a, which clustering can solve the classification problem, but I don't know how many classes that you would have here which would be able to do that. In clustering, if there's not two clear clusters, you might have issues there. So have you considered other approaches?
We have considered other approaches, but we've been kind of practicing more with the clustering as of right now. And so, like, as we progress into the research, then we're open to any other forms of analysis.
Okay, excellent job. This is fantastic.
Okay, awesome. Okay, thank you to all the presenters. You guys did awesome. I loved hearing your research. And this will formally conclude the Computer Science Division